Okay, so good evening. This is your week 4 session. So in this week's session, we will be talking about the X-ray crystallography, in which you have gone through the production of X-rays, their properties, what are unit cell, what is symmetry, why it is important, what are crystal lattice, and what are asymmetric unit, and different concepts about the crystals, crystal systems, and why we should understand them. And then instrumentation, which is used in X-ray crystallography. And then you have also looked into the data collection process and how further refinement is done. So with this, we'll practice few MCQs, and then we'll also look into the current literature, that what are the latest reviews saying, and then we'll be talking about your doubts. Okay, so let's begin with the production of X-ray and their properties. So for the production of X-rays, mostly the power is supplied towards the X-ray tube via the cables. So milliamperage heats the filament in the cathode, producing the electrons. So as soon as the electrons are produced, because the current is flowing, heat is generated. So now as soon as electrons are produced, so we supply positive voltage through the anode. So now anode is positively charged and electrons are generated at the cathode. So electrons will move towards the anode because it is positively charged. So as soon as the electrons start moving, firstly their speed is nearly half of the speed of light, but as soon as they reaches closer to the anode, their speed means slows down. So they slows down, and then as soon as they hit the anode, there is generation of heat, which is 99%, and 1% X-rays are produced. So this is the way how the X-rays are produced. So in this figure also I have shown that how from cathode electrons are moving in projectile motion, hitting some target which is our anode, positively charged, and then some heat is dissipated and 1% of X-ray beam is produced. Now we will talk about the tube interactions and what are the properties of these X-rays. So as we all know that X-rays are the electromagnetic radiations, and they have wavelength between 0.01 nanometers to 10 nanometers. So this is the range in which the X-rays have the wavelength, and typical X-ray diffraction wavelength is 0.05 nanometers to 0.25 nanometers. And in the previous sessions also we have discussed that why we need this typical wavelength, because we want when we want to study any particle or any macromolecule, so the wavelength should be less than the length of that particular molecule or size of that particular molecule, then only we'll be able to visualize it. So that's why this is the typical wavelength of X-ray diffraction. Now comes the X-ray generation system. So it is compact and sealed, eliminating the need for vacuum pumps. So in the later slides, we'll see that what are the different X-ray generation systems, but it is hoped that it should be compact and it should be sealed so that there is no interference with the external environment, and the tube is water cooled. So as we have seen that whenever those electrons are moving, so they are dissipating some heat. So to avoid that, we use the water as a coolant. So to dissipate that heat, um, then later on we will also see that how our different techniques have come up, different instruments have come up, and how they deal with this heat generation. So previously water was used as a coolant, but later on different other techniques have been opted. So with which we means deal with that heat generation. Okay, so this is the same means old X-ray tube, that it flows some water to deal with the heat generated, and then how the voltage is supplied, and then X-ray beam is produced. So for more detail, we'll move to the next slide. Okay, so now we'll talk about different X-ray sources which are generating X-rays. So there were early X-ray sources such as Crookes tube, which was also known as cold cathode tube. So it was used in early 1900s, and it was relied on the ionization of residual air. So in this tube, this is the glass tube, also known as discharge tube. So in this, there is air which is at low pressure, and then this is the cathode from where the charge is supplied. So negative voltage is provided, and now it will generate some electrons which will try to move towards the anode, which is having the positive charge. So it consisted of a glass bulb with low atmospheric pressure gas, and it is unreliable as the residual air which is present inside this tube, it got absorbed over time, so which leads to its failure. So that's why we means in this case we have also need to maintain the vacuum, and then uh, this is the disadvantage of this particular, because after some time air will get absorbed, so we need to supply the means more air. So that thing was difficult. So that's why we moved to the latest X-ray sources. So means to avoid the air, so we use the sealed tubes. So in this case, the entire X-ray generation system is sealed into a single unit. That's why it eliminates the need for the vacuum pumps and motors to spin the target. So this tube is water cooled to dissipate heat. So in this is also the older one only, in which the water was used as a coolant, and lower power was supplied so that overheating could be avoided. Because if power will be high, then the electron will move with greater speed, and then it will generate more heat. So in this case, to deal with it, they provided lower power. So due to lower power, you will not be able to get the high speed. So this was also one of the limitation also, because you have to deal with overheating as well. So this is a self-contained system, and its maintenance was easier than the previous cold cathode tubes. So this is how it looks. And in this case, because it is a steel tube, so we do not require the vacuum inlet as it was used in the previous case. So this is the glass and steel body. So as you can see, this is the steel body, and then the same mechanism is working here also, that external voltage is provided, and from the cathode, the electrons will be generated. There will be a filament which will heat up and generate the electrons, and then those electrons will move towards anode, which is positively charged. They will hit the anode, and then X-rays will be generated. So the process is same, but the body, you can say, is different. So the technique is different of generation. And then the latest, which is most commonly used X-ray source, that is rotating anodes. So in this case, the anode is spinning, because in the previous also, as we have seen, that electrons are moving with high velocity from the cathode towards anode. So as soon as they hit the anode, there is some kind of heat generated, and that anode is also getting degraded with time because it is getting hit. So in this case, as the anode is rotating, so we can you can say we can deal with that damage which is means happened due to the striking of the electrons. So in this case, anode will spin at 6000 rpm, which spread the heat over the larger area. So in this, we will also be able to deal with the heat which is generated, because previously it was fixed. So heat is generated only at the smaller area where the heating was happening. So now in this case, it is moving. So now heat is spread across the larger area, and it requires maintenance because of the wear and tear on the bearings. So bearings are these things which are helping, which are at the means rotator part. So they are helping in the rotation of this anode disk. So with some time, because they are continuously rotating the anode, so with some time these bearings can get wear or tear. So this maintenance is means it require high maintenance because we have to deal with that. But otherwise, it can deal well with the heat. And then typical anode materials are tungsten rhenium on the molybdenum core, which is backed with graphite. So these are the choice of metals which are used. And then they can withstand temperature up to 2500 degrees centigrade. So as we have seen in the previous case, because heat was generated, so it was using only lower voltage. But now in this case, as the anode material can withstand that much, means this much amount of heat, so now we can generate X-rays with higher intensities, and it will be faster. Generation process will be faster. And example of such rotating anode X-ray source is Rigaku generator, which is most commonly used, and it is also known as home source. So home source is the source with which the X-rays are generated and which is most commonly used. So mostly Rigaku generator is the home source because it generates high brilliance X-rays. Okay, so now I will talk about the little bit of a difference between the rotating anode X-ray diffraction versus sealed tube. So sealed tube was the one which we have seen at the second time. So in the sealed tube, as we know that it is sealed inside a body in which water is provided as a coolant to deal with the heat which is generated, and it generates means this generator is rated to 3 kilowatt, and sealed tube is rated to 2.2 kilowatt. So this is the limit with which to which they can you can say uh, means how much heat they can tolerate. So actual operating power is 40 kilovolt and 40 milliampere, which sums up to, or which is approximately 1.6 kilowatt. So this is very low. So that's why they use low power only. They cannot deal with the high power. But if we talk about the rotating anode X-ray diffraction, so in this case, as the anode is rotating, so heat is spread across the larger area, so we do not have to worry about that. So in this case, the generator is rated to 9 kilowatt. so which is approximately 3 times more than the sealed tube X-ray diffraction. And then this rotating anode is also rated to 9 kilowatt, and the actual operating power is 9 kilowatt. So which is 5.6 times greater than the X-ray flux compared to the sealed tube. So that's why rotating anode is much preferable than the sealed tube X-ray diffraction method. And then comes synchrotrons. So these are also the type of X-ray sources. So means after the home source, the synchrotrons have come up. So these are the large tubular rings which are under high vacuum. So where particles are moving at the speed of near light. And the bending magnets generate intense X-rays. So in this system, to generate or intensify the X-rays, there are some bending magnets. So as you can see in this figure, so this is just a representation. There are multiple places, as you can see, whenever the X-ray will be moving. So there are bending magnets. At each level, they will increase the momentum of X-rays. So with this, the intensive means the X-rays are intensified at each level. So that's why they generate the X-rays which are with high intensity. And then insertion devices like wigglers and undulators. So these are also some other insertion devices which are add-on with the synchrotrons, and they further enhance the brightness of X-rays. So these are the latest, you can say, sources which are generally used. But the limitation is that they require a larger area where these synchrotrons need to be set up. So these means there are certain so in the next slide, we'll see that there are certain places where these synchrotrons have already been installed. So we have to go there to study with these types of X-rays. So this is just a slide for your, means if you want to study in detail that what each and every part is doing. So you can go through this slide for details, and I will not go through them. So now these are the notable synchrotrons which are worldwide present. So in USA, there is NSLS Brookhaven, APS chess, ALS, SSRL, and then in other countries also there are certain facilities where you get the synchrotrons. So among all of them, the APS synchrotrons beamlines, they are 1,000 times brighter than the home sources, which is Rigaku generators. So that's why they are much preferable because they are much brighter, much intensity of X-ray can be obtained from them. So that's why they are much preferred. And this is just the representation. So this is the place. This is the picture of this APS, and this shows how the synchrotron is present. So means from the source the X-rays will be produced, and then at each level there are bending magnets. So these are some bending magnets which are present. So at the time when that X-ray particle will reach the bending magnet, so that magnet will intensify X-rays at each level. So that's why intense, or you can say brighter X-rays are produced. So this is the logic behind the synchrotrons. Yeah, and in this also I'm showing the same, that particles from a pre-accelerator, which is LINAC or smaller synchrotron, is coming, and then it is reaching this particular magnet, accelerating cavities working like mini LINACs. A magnetic field bends the beam. So with the magnetic field, there is bending of beam is happening. So beam is X-ray beam which is coming, and then it is reaching the target and collision detectors. So this target and collision detectors will be present at the end after the high intensity beam is obtained. Okay, so now we'll now talk about the collimators. So this was all about the X-ray sources, that how the X-rays are generated. So is it clear till now? Yeah, okay. So now we'll talk about the collimators. So collimator is also a device which narrow down the X-ray beam to focus it in a specific direction. So now as we have generated the X-rays, so now they are coming, they are not focused in one specific direction. So to do that, we provide this collimator in which these are the parallel metal plates which are closely packed, and there is only a smaller gap in between them. So what will happen that as soon as X-rays will come, only the X-rays which are passing in a specific direction, only they will pass. Other X-rays which are like scattered kind of X-rays, they will get absorbed by these metal plates which are present. So in this way, this is helping to narrow down the X-ray beam, and then it is also providing a specific direction to those X-rays so that they can properly pass through the crystal. So this is the representation of the collimator, that how it is present. So in this case, as you can see, the X-rays are generated. So this is for the sake of representation only, that From this cathode, the electrons are generated, moving to anode, hitting the anode, heat is dissipated, X-ray is generated, then X-ray is passing. And then there is a collimator. What it is doing, it is only passing the, means it is narrowing that X-ray beam and passing them to the specific direction. Now when it will pass, means towards the crystal, so there will be another devices which goniometer will be present, which will be holding the crystal. So now it will pass from the center of the crystal and get diffracted back. So these are the detectors which will catch that diffracted X-rays, and then other processes will happen. So that we will see in the later slides. So this is the work of collimator. Then comes monochromator. So it transmits a narrow band of selected wavelengths. And the types of monochromator, so as the name suggests, monochromator, so it is a device. Mono means single, chromator means color. So as you can say, it means from a multiple colors, it only passes the single color, or you can say it passes only a specific wavelength of light. So this is what the monochromator is used for. So there are other interference filters which utilize the X-ray absorption edges of the elements, and there are some crystal monochromators. So these interference filters, they are like add-on to monochromator, or you can use them in place of monochromators, because these filters, they will only allow particular wavelength to pass. So for example, in these week 4 sessions also, as you have seen, that there is generation of K alpha and K beta different light radiations. So out of them, only K alpha is allowed to pass because it is of high intensity, and K beta is not allowed to pass. So in that way, these filters are also filtering out the K beta and then only allowing the K alpha intensities to be captured. So monochromators also does the same work. It only allows specific, or you can say narrow band of selected wavelengths to pass through them. And advantages is that it will improve the resolution, it will reduce the background radiations, and it will also enhance the signal to noise ratio. So in this, as it is showing, this is just for the representation sake, that different colored wavelengths are coming and only this purple color light is allowed to pass through it. So this is the work of the monochromator, only allow a specific wavelength of light to pass through it. And then ultimately, there is a role of X-ray detectors, because now X-rays have passed, they have passed to the crystals. So now X-ray detectors will detect those X-rays which come after diffraction from the crystal. So these devices measure the X-ray flux and their spatial distribution as well as intensities. So the categories of detectors are imaging detectors, which were previously used, such as photographic films or photographic plates, films, and digital detectors. So this is one of the figures showing the photographic film, and this is showing that how the means diffracted lights are coming. Then we need to generate the reciprocal plane, and then from the reciprocal plane they are reaching the photographic film, from where the diffraction pattern is studied, and then models are generated, that what is the distribution of atoms inside that particular crystal. So then comes counter methods. So imaging detectors were previous techniques, and then latest techniques are the counter methods, which measures the X-ray dose, that what is the X-ray dose which is being generated. So first of the counter method is the Geiger-Müller tube. So Geiger-Müller tube is the tube which is generally filled with the inert gas, and in many cases it's argon, and it has a central anode which is at 800 to 1500 volt. Then electrons accelerate, ionize the argon atoms, and create an electron avalanche towards the anode. So in this case, what we are counting the intensity of pulse which is getting generated, and how that pulse is getting generated, because now the X-rays which are getting diffracted back, now they are coming to this tube. This tube is collecting it. Now this tube is already filled with argon. So it depends that at what intensity that X-rays are getting diffracted. So with that intensity, they will hit these inert gas molecules, and then they will ionize these argon atoms. So after ionization, because it is getting hit by the electrons, it will get ionized and it will create an avalanche of electrons. So multiple electrons will be produced, and now these electrons, they will tend to move towards the anode. So anode is the middle positive plate which is positively charged, and electrons are negatively charged. So they will move from this end, which is the negative electrode. So these edges, you can say edges of the tube or the boundaries of the tube, that is negatively charged. From where the electrons are moving towards the central anode, they will hit the anode, and now this particular anode, with it is attached to the counter, and it will capture that it with which intensity the pulse is generated. So this is how it is telling you that what is the dose of X-ray which is it is receiving. So this is the counter method, and this is the means this is how small this Geiger-Müller tube actually looks. So second counter method is the proportional counter. So this is similar to the Geiger-Müller, but the difference only lies that it, instead of argon, it has the heavy gases which are easily ionizable, such as xenon and krypton, and it operates below the Geiger-Müller plateau. So this is the plateau, this is the voltage. So it need to be operated at this particular voltage, but as the proportional counter, so it is operated generally between this 200 to about 580 voltage. So 580 volt. So this is the power at which it need to be generated, because it has the easily ionizable heavy gases. So that is also one of the reason, and it has a short dead time, which is about zero point 0.2 microseconds, and it accurately counts the high rates. So that's why it is the another method, alternate method you can say, to Geiger-Müller if you want to operate at the lesser voltage to that of Geiger-Müller. And this shows that how the radiations are coming. This is the window, and central anode is positively charged. So as soon as the radiation will enter, they will try to ionize the xenon or krypton gas, which is filled inside the tube. That gas will get ionized, it will generate electrons. All those electrons will come to anode, they will bind the anode, and then because the electrons are means attaching to anode, they will generate some pulse of, so some pulse will be generated which will be captured. So that pulse will tell you what is the dosage of the X-ray radiations, because if the X-ray radiations are greater, or they are of high intensity, then the pulse will be stronger, otherwise pulse will be weaker. So in that way they help to count the dosage, and this is how the proportional counter looks like. So these are just the small cylindrical tubes. Then comes scintillation detector. So this is also another counter method which uses the thallium activated sodium iodide crystals. So instead of heavier gases or inert gases, they uses the thallium activated sodium iodide crystals to emit the visible light when struck by X-rays. So in this case, X-rays will come and they will hit the thallium activated crystal, and as soon as they hit the crystals, it will be generated, means they will emit the visible light. So visible light will be generated, which will be detected by the photomultiplier tube, and it measures the shorter wavelength X-rays as well. So in this way they are generating a visible light which will be captured. So previous case, the electron or the pulse which was generated, whereas in this case the visible light intensity that will be captured, and that will tell you that what is the dosage of the X-ray radiations which are hitting this particular thallium activated crystal. So other crystals include anthracene, naphthalene, and p-terphenol. So there are different choices of crystals depending on the light at what means how they generate visible light. So based on that, we can use different types of crystals. Now this shows that high energy photon is coming, and these are the low energy photons. So means high energy photon will generate the greater intensity of visible light, means greater energy, and these are the primary electrons which are generated. Now when primary electrons further hit something, then there is generation of secondary electrons, and in this case they are showing that how the multiple electrons have been generated, and ultimately connector pins are there, and in the end this connector pin will be attached to a detector which will carry the what intensity the visible light is produced. So this is how they measure the, means whether the X-rays which are coming, they are of higher energy, lower energy, what is the dosage. And then there are semiconductor devices. So now in this case, they convert the X-ray signals into electric pulses, and in a silicon lithium drift detector. So there is a silicon lithium drift detector, and X-rays generates the electron hole pairs. So as you can see in this, that these blue are the electrons and these red ones are the holes. So whenever the electron will come, because there is a silicon lithium drift detector, so X-rays, they will generate, they will break down, they will generate electrons as well as holes. And those holes, because they will, electrons will be negatively charged, and these holes will be kind of a positively charged, so they will move towards the negative terminal, whereas electrons, as they are negatively charged, so they will move towards the positive terminal, or you can say towards the anode. So electrons will move towards the anode, and holes will move towards the cathode under the voltage, which will produce a pulse. And this pulse voltage, which will be calculated V equals to Q by C. It will tell you total charge collected on electrode divided by C, which is the detector capacity, measures the X-ray intensity. So with this, you will be able to capture the what is the X-ray intensity. So by changing the formula, the pulse which is generated with which you will be able to identify that what is the X-ray intensity. So these are different methods with which we can count the dosage of X-rays. And with this, we won't be able to distinguish among different uh, atomic positions, but they tell us the means at what level the X-rays are generated. So these are only for the X-ray dosage and means they cannot tell us about the you can say these are only at the level of intensity and not at the phase level. So in the later sessions, we'll see that how we can deal with the phase problem, that at what angles the light has been diffracted. So then comes X-ray diffractometer. So this is the sum up of all the things which we have seen so far. So X-ray diffractometer is the device which measures the diffraction pattern of X-rays which are interacting with the crystal. So its components are X-ray source, so which is very important because it is generating the X-rays. Then comes collimator. So this also we have seen that how it only allows the specific, it narrows down the or it provides a specific direction to those X-rays. And then there is a sample holder. So sample holder is the goniometer which is holding the particular crystals at in proper orientation, and then it is also rotating them in different directions so that the X-rays can properly pass through the center of the crystal and proper diffraction pattern could be achieved. And then there is a detector. So in this case, they have not mentioned monochromator, but monochromator is also the important part. So after collimator, there is also a monochromator so that a specific wavelength of X-rays could pass through the sample. And then ultimately detector will detect the X-rays which have been diffracted. Yeah, so this is the basic instrumentation, that electric power is coming, voltage stabilizer is there, high voltage generator, and then X-ray, from X-ray tube the X-rays are generated, the electrons which are generated, so they are means producing the X-rays. X-rays are passing through collimator and then they are reaching the crystal. They will hit it and then diffraction pattern will be generated, which is captured by the detector. And now the detector, after capturing it, so now detector will pass it to amplifier, which will amplify the waves which have been obtained. So amplifier will amplify it, then there is scaler, and then ultimately printer. So printer will print the pattern and scaler will capture that at what are the distances between those atomic particles, you can say, inside the crystal. And then there is a rate meter and the recorder. So this is also capturing that at what rate the diffract means ray is received. So in this way, this is the basic instrumentation of a diffractometer. And till now, we have only studied till detector, and the later thing about the data analysis, indexing, and all those things, they will be covered in the later part. So I hope, is it clear till now? Okay, so you don't have to worry about the proper details of each and every method. It's just for your understanding that what are the different tools which are used. So you need only to get the basic understanding of these things, that why they are used and what are the means little bit about their background, that why they are used and how they are different from each other. So now we'll talk about the X-ray emission and its absorption, because means why we are studying what we are getting. So towards that, so X-rays, so this is the characteristic radiation which is produced when an electron transition occurs between the inner atomic shells. And then comes Bremsstrahlung radiation. So this is a continuum radiation which is emitted when electrons are decelerated upon impact with the anode. So as you know that in the entire process of generation of X-rays, we have seen that how the electrons are coming from cathode and they are hitting the anode. So as soon as those electrons they hit the anode, so that anode has a metal or there is some element which is present at the surface of the anode. So where those electrons are hitting, and now because they are coming with a greater acceleration, they are coming with higher speed. So as soon as they hit, they get decelerated. That means their speed drastically reduce. So with that, after hitting, there is a generation of some radiation. So that is known as this Bremsstrahlung radiation. because as soon as they hit, they slows down, and then something means when some radiation, continuum radiation is produced, that is known as the Bremsstrahlung radiation. Yeah, so in this case, I have shown the relative intensity versus the photon energy. So this is, as you can see, this is the higher means greater the voltage which is provided, greater will be the relative intensity with which the radiations will be produced. And now this shows that what is actually happening at the atomic level. So what electron is actually doing. So what it is doing, that in the anode, the filament, metal filament which is present on the anode, so that high moving electron will come and it will hit the, if we talk about the atomic shell of the particular atom, so now what it will do, it will hit that particular, at different, there are different levels, you can say different shells, so it depends that at which shell that particular electron will hit and move that particular electron out. So in this case, if the kicked out electron is from the K shell, then it will generate some different radiation, or different radiation will be generated, because as soon as they will kick out this electron, so what will happen now, another electron which is present in the outer shell, it will try to occupy the space of this electron which is kicked out. So when the electron which is present in the L shell, when it moves to the K shell, after the K shell electron getting kicked out, so this type of radiation which is produced, that is known as K alpha. And if the electron is coming from M shell directly to the K shell, replacing that electron, because the electron from K shell is kicked out. So when M shell electron will occupy the K shell, so the radiation which will be generated after this movement, it will be K beta. And then there is also L alpha, when the electron from L shell is kicked out and from M shell electron is coming to occupy the place of this L shell electron. So that is known as L alpha. And always keep this thing in mind, that electron won't go from inner shell to outer shell when the electron is kicked out. So that's why the movement will only happen towards the inside. So from M shell to L shell, and from L shell to K shell, or from M to directly to K shell. So that will be K beta. So in this case, in X-ray diffraction, we are mostly interested in the K alpha kind of radiations because they are of high intensity. K beta are slightly lower in intensity. So yeah, so in this case, as I have shown the intensity ratios, so K alpha is higher intensity, K alpha 1, and then K alpha 2. Even means it also depend on the orbitals also. So K alpha 1 to K alpha 2 to K beta, if we calculate the intensity ratios, then K alpha 1 is having the highest intensity, which is 100, and then K alpha 2 50, and K beta is 20 if we calculate their ratios. So the best intensity will be K alpha 1, in which the electron is coming from the last orbital of the L shell. So K absorption, so means this radiation which is being generated. So this is K absorption, also known as K absorption, the rays which are, the radiations which are produced, K alpha 1, K alpha 2, and K beta. So if we talk about copper, it is commonly used in protein crystallography. So now we'll see that at what means at what intensity it generates the K alpha radiation. So its intensity is 1.54060 angstrom. So means this is the wavelength, sorry. So this is the wavelength. And then for molybdenum, it is 0.709. And this molybdenum is used for the smaller molecules, because we already know that this wavelength, it dictates that what size of molecule you can study. So as copper has slightly greater wavelength as compared to molybdenum, so that's why molybdenum is used for smaller molecules, whereas copper is mostly used for the protein crystallographic studies. And this shows the respective K alpha and K beta intensities of both of copper and molybdenum. So for copper, the absorption is 0.154 nanometers, or you can say 1.54 angstrom. And in case of molybdenum, K alpha is zero point 0.7 angstrom, or you can also call it as the 7 nanometers, zero point 0.07 nanometers. Okay, so now so now I hope it will be clear till now that why we are studying K alpha. Now we will talk about the X-ray diffraction. So Bragg's law and all those things. Now we'll cover that. So Max von Laue was a scientist And in 1912, he experimentally means X-ray diffraction was developed by him and also validated by another two people who were working under him, such as Frederick and Knipping. So Bragg gave a law in 1913. So this was also a father-son pair, William Henry Bragg and William Lawrence Bragg. So they said that n lambda means n times the wavelength of light is equals to 2d sine theta. So where n is an integer, lambda is the wavelength of X-ray, d is the interplanar spacing because now we are talking about the crystals, and theta is the diffraction angle. So whenever incident light hits the particle, which is the lattice point, or you can say in the unit cell, there are different particles or atoms. So as soon as the X-ray will hit the uh, means this particular particle, so it will get diffracted at some angle. So this particular angle, the angle at which it get diffracted, that is theta, which is the diffraction angle, which is made with the plane. So this will be the angle theta, and incoming radiations will also make some angle with the plane, so that will also be theta. So both of them will be theta. So this is the diffraction angle, and it is used for determining the crystal structures. Okay, so now let's see in this figure that how the incident beam is coming, and this is the A, which is the point. So these are telling you the, if we talk about proteins, then these are the smaller atoms which are present in the protein, and then multiple atoms will be present, and ultimately what is our aim? To see the distribution of those atoms, that at which point, which is the coordinate position of those atoms. So we want to capture it with X-ray diffraction. So now X-ray beam is coming, and it will hit those particular atomic points, and then it will get diffracted. So in this way, these are the two unit planes, and this is the distance between two planes, which is generally d. And now our aim is to find the path difference. So what is the path difference? So in this case, n lambda 2d sine theta, what we are doing is, lambda is the wavelength. Wavelength means what is the length of a single wave. So that is lambda. When we say n lambda means n is the integer number. So it depends that for how many waves we are calculating the length. So in this case, if we need to find the length of two waves, so it will become 2 lambda, 2 times lambda. Sorry, lambda plus lambda. So it will be 2 lambda. So that is how we calculate the wavelength. Now, how we will find this wavelength? So among two waves, the length will be CB plus BD. So which is CB, BD. So what is this length? If we apply the mathematical trigonometric calculations, so we will know that to find this, as we know the theta angle is made with this plane, and if we talk about the mathematical calculations, then we know that sine theta is equals to perpendicular over hypotenuse. So according to this, if this is the perpendicular and this is the theta, so CB or BC will be a hypotenuse. So we know that the perpendicular which is made, so perpendicular length is D, and this is the hypotenuse. So with that formula, lambda will be equals to this, BD will be equals to D sine theta, because it is the hypotenuse. So it will be D sine theta. Similarly, it will be D sine theta for BC as well. So if we add CBBD, then it will become 2D sine theta. So this is how with mathematics they have calculated it. So in this way, the Bragg has identified that what will be the n lambda. Yeah, so this is how the Bragg's equation works. Now we will talk about the crystal lattices, that what are the different crystals, which means what are the different types of crystals and what are actually crystals, and what a lattice means, what are these points and how we denote them. So lattice, when we say lattice, it is a 3D arrangement of points. And what are those points? So in proteins, different atoms, they are the points. So points in the space that represents the structure of a crystal, and they defines that how the crystal's internal patterns repeat in all directions. So in this case, uh, these are the lattice points, and the smallest unit is the unit cell, which combines together to form a bigger crystal, or you can say bigger crystal lattice. So lattice is just the three-dimensional arrangement, and whenever you means stand on a point in the crystal lattice, so you will see all other points, they will be at equal distance from this particular uh, point or lattice point. So in a crystal lattice, each point from the let means each every single point will be at the same distance from the lattice point, which is generally D. And then comes the concept of asymmetric unit. So this is the unit cell, and asymmetric unit is even the smallest part. So it is also smaller than the unit cell. So it is the smallest part of the crystal structure, which is repeated through symmetry operations. So with symmetry, we are repeating the smallest unit, and then they compose the unit cell. And multiple unit cell, they compose the lattice. So with different asymmetric unit is the smallest unit, which is repeated through symmetry operations, and it will form a unit cell. So it contains the unique structural information which is needed to describe the whole crystal. So means this one will be the smallest one, which is unique, and the other things which will be present, they will be just the symmetry operation of that particular asymmetric unit. And then comes unit cell. So this is the basic building block of a crystal. It is small, repeating box-like structure that when stacked in 3 dimensions, it forms an entire crystal. And a crystal can contain one or more asymmetric units. So I'll give you a little bit of example. So for example, in a protein, if we only talk about the monomeric protein, so in monomeric protein, which has only a single chain, there are motifs. So when we were talking about the secondary structures, then we have seen that there are certain motifs which are present in repetitions in a protein. For example, alpha, beta hairpin. So there are multiple motifs which keep on repeating. And even the alpha and beta helices, those secondary structures, they are also getting repeated after some time. So if we talk about a protein, so in that case, the motif, if only a single motif is present in the entire protein, so in that scenario, motif will become our asymmetric unit, which is only getting, means it's only the symmetric operations are performed and it is arranged in different symmetries to one another. So it will compose the asymmetric unit, and there could be multiple motifs in a single unit cell. So in this case, we have two motifs, and those motifs are only varying in their symmetric operations. So if some symmetric operation is performed on one of the asymmetric unit, so with means their variation is only at the level of the symmetric operations which are performed. And now these unit cells, they will stand on each other, they will get stacked, and they will generate a crystal lattice. So in this way, crystal is a solid material with regular repeating atomic arrangements, and it is composed of many unit cells which are stacked together in a precise order and which will form a well-defined structure. Is there any doubt? Okay, thank you. Yeah, so this is how, what means, I hope now it is clear that what is asymmetric unit, what is unit cell, and how different unit cell combine together to form a crystal lattice. So lattice is just a term which is used to define the 3D arrangement, and crystal is the outcome, the structure which is formed. So now why the crystal symmetry is important in data processing? So knowing the symmetry, it reduces the asymmetric unit size and data collection time. Because now, as I have said, that if you know exactly what are the smallest repeating units, so you do not have to perform multiple operations. If you get a smaller means, for if asymmetric unit, you get the idea that how the atoms are arranged, then by performing symmetric operations, you can get the idea that how the other units will be arranged. So you do not have to go through a high level of calculations. You will know that what are the rotations which you need to perform. So in that way, this if you know the asymmetric unit, then you can save your time and efforts as well. So now there is an international tables for crystallography which define the crystal symmetries. And then crystal lattice, it has a long range ordered structure with repeating units. So why we are again and again focusing on the repeating units? Because we do not want that we waste our time performing the same calculation, because there is a redundancy in the structures as well as in the crystal also. There are multiple proteins which are aggregating together to form that crystal. Not, we can't say the aggregation word, but they have the means supersaturation has happened. So in this way, we want to minimize our efforts and maximize the output which we want to generate. So for that case, we must know that what is the symmetry of those crystals. so that we can know that what are the fold x's which are possible. Means after how many rotations will they generate the same, they will reach to the similar position, so that we can define that with the goniometer as we are moving the crystal. So we will be knowing that at what angles we need to rotate it and capturing the diffraction pattern to avoid the redundancy simply. So this is the representation that how do we identify that what will be the lattice, what is valid lattice. So in this case, if we talk about these two smaller unit cell, so these are not, these are different. So we cannot call it as the smaller unit. Or you can say, if we talk about in terms of asymmetric units, so this is not the smallest unit which is identical. These both are different. So we cannot call them as valid lattice. So now what will be valid lattice? So now we need to increase the size. So if we encode all of them together into a single unit cell, or you can say single asymmetric unit in this case. So they both are, means the boxes which are highlighted, these are actually square boxes only. So this shows that these are the repeats. So in this way, these are identical to each other. So that's why they are the valid lattice and basis, because they repeats exactly. Whereas in this case, these repeats are non-identical. So whenever we define lattice, we also define that they are identical repeats. So if they are varying, then that is not the true lattice. So this is how the lattice are defined. Okay, so now we'll talk about different space lattices. So when we say space lattice, it means that the array of points where every point has identical surroundings. And in Euclidean space lattices, they are in infinite array and have translational periodicity. So in this case, as they mentioned, that every point has identical surroundings. So each time whenever you, means if you reach at this point, so it will have a point towards its left and towards its right also. So similarly, after reaching every terminal point, you need some other point to be, to means succeed it. So in that way, there will be infinite points or infinite lattice points which will be present in the space lattice, and they will be moving continuously. There will be translational periodicity. So translation means the movement. So that will be periodic movement. This distance will be a, which will be means constant. So the distance between each lattice point will be constant, and there will be infinite data points on both the sides to maintain the identical surrounding on both the sides. So when we talk about 1D lattice, so this is the example of 1D lattice. So its properties are infinite translational periodicity in Euclidean space. So this is means this defines that what is the space between each of the lattice point, and they are described by the single lattice parameter, which is A. And there is symmetry in these one-dimensional lattices, because if we means look at the mirror images of two lattice points, they will be identical. If we perform 2D rotation, then also they will be identical, and even if we perform their inversion, then also they will appear identical. There will be no difference between the inverted and the non-inverted form. So this shows that this is means only a single point. It is just a single point, so that's why it is completely symmetric. And a line segment of length A is the unit cell in case of 1D lattice. So this is also known as line length, where we are only looking at the line because it is one dimensional. So then we move to 2D lattice, but before that, the linear Bravais lattice. So Bravais was a scientist who has defined 14 different lattices. So he has mentioned about the linear Bravais lattices, that one dimension, so only one vector. So that vector is A, which is the distance between the two lattice points. And this vector has no length or angle relationship to other vectors. So there is only one possible Bravais lattice, and this shows that how there is translational periodicity. So in this case, because we wanted in lattice we should have identical points which are repeating, so that's why these all will combine to form a Bravais lattices don't show mirror symmetry, only translations. So which means that um, they are like moving forward in space. So means there is some translation. So in the later slides, I'll show you the example of translation, what it actually means. So translation is just a movement which is happening at some distance. So they don't show mirror symmetry and uh, only translation. And this unit of six circles, which are known as the bases. can be collapsed into a single point, which is known as a lattice point, which repeats at a distance of a lattice vector. So as in this case, it was A. Okay, so means each lattice point can be composed of bases. So bases can also be called as a asymmetric unit in other terms. So this is about the 1D lattice, which is not of much importance in our case. So our important is the 3D lattices, which is actually the three-dimensional structures. So secondly, we'll talk about the 2D lattices, in which now previously there was only a single line. Now in this case, there will be two basis vectors. So previously it was only a vector. Now there will be two vectors, and which are described by three lattice parameters. So now we are moving from a line land to a flat land. So there are two distances which are A and B. So A and B, and then there will be one angle between them which will be alpha. So now there are four possible unit cell shapes which will lead to five lattice types. So now when we consider two different vectors, so there is a possibility of five types of lattices: square, rectangle, centered rectangle, 120-degree rhombus, and parallelogram. So one by one, we'll go through them, that means how they are produced and what it means. So this is just a table to show you their symmetry and what is their shape of unit cell and the lattice parameters such as A and B and then alpha angle. And then there is also one of the example that as we have considered rectangle and centered rectangle, so why not centered square? So this is just a question in which they have shown that if we consider a new point in the middle of the unit cell, then what will happen is that from these two middle points, we need to construct another, because centered square lattice will have the points in the center. So these are the lattice points in the center. We need to join these four lattice points, and again the structure, or you can say the symmetry which it will be generating, that will be identical to that of the square. The difference will only be the length of the A and B. So these A and A will again be equal to B only, and the angle which will be formed, it will also be again 90 degrees. So there is no variation. So that's why we do not have centered square lattice, whereas we have centered rectangle lattice. So now we'll look into them in detail. So if we talk about squared lattice, as I've discussed, that this angle theta, or you can call it as alpha, this is just the name. So it will always be 90 degrees, and the distance between two lattice points will be identical if we talk about the square lattice. Now we'll talk about the rectangular lattice. So as we know about the shapes also, so in rectangle, we do not have the, means both the sides equal. So in this case, A will not be equals to B. Whereas if we talk about the angle alpha or theta, it will be 90 degrees. So this is also the another lattice, because in this case, now we will also be talking about the, because there are multiple layers of the, these are the layers where lattice points are arranged. So in this case, there are multiple layers. They, in each layer, these lattice points, they are equidistant. But what about the other layers which are lying above them? So in that case, this distance can vary, because now B is also coming into picture. So when we were talking about the line land, or you can say, yeah, in the line land, we were only, because there was only a single layer. So in that case, we were only worried about A. But in this case, we are also worried about B, because among two different layers, their length could vary. But among two layers, the lattice points in each of the layers, so their distance will be, they will be equidistant. So this is how the rectangular lattice can also come into picture. In other scenarios, there is a possibility that both these A and B points can also be equal. So that's why square lattice is also possible in 2D lattices. So in this case, we are just looking for possibilities, that what are the possible lattices which can be generated. So this is the rectangular lattice. Now we'll talk about the, yeah, so in rectangular also, if we consider the, it is also, it could also generate the centered rhombic lattice. So for example, if we consider a rhombus, so this is the rhombus. In case of, means instead of talking about this rectangular, if we consider in terms of rhombus, then there is a point in the center. So in the later we'll see what the centered means. So centered means that after constructing this smaller unit cell or the smaller plane, there is a point which is lying in the center of the plane. So at that time we'll call it as a centered rhombic or centered rectangular, because the plane which we'll be considering, it will be having the point in its center. So in this case, if we talk about centered rhombic lattice, so this will be means this is as means it is also generating from the rectangular lattice only. So means if a rectangular lattice is present, or in the crystal if it is a rectangular lattice, so in that case there is a possibility that we can also generate the centered rhombic lattice. So it is just on us that how we see it. Some people might see it as a rectangular lattice, whereas others can construct it in a form of a rhombic lattice. So this is just to show you that how different people can perceive it. This is also fine. In this case, A will be equal to B, but angle theta won't be equal to 90, which was previously 90 in case of the rectangular lattice. Now we'll talk about the centered rectangular lattice. So as I have mentioned that whenever we'll be talking about centered something, so it means there in that plane which we are considering, there will be some lattice point which will be present in the center of that. And this centered rectangular lattice, in some textbook, it is also referred to as orthorhombic. So in this case, the A will not be equal to B because it is a rectangle. So both the sides will be of different length. But the angle which will be formed, that will be 90 degrees. So it has an extra lattice point in the center. This is only the difference among the rectangular lattice and the centered rectangular lattice. Yeah, and in this case, centered rectangular lattices are also called orthorhombic. So why orthorhombic? So because if we take two center points of a centered rectangular lattice and combine them, means we are now shortening them. So what is other perception for centered rectangular lattice? That we can also call it as the orthorhombic. We can also design the orthorhombic plane in which A will be equals to B, but theta is not equal to 90 degrees. So this is the another way in which centered rectangular lattice can be perceived. Yeah, so this is the conventional cell and this is the primitive cell. So primitive, now I'll just tell you a short difference between the primitive cell and the conventional cell. So conventional cell is just written because by convention we consider centered rectangular lattice in which the lattice point is lying in the center. But why it is called primitive? So in primitive cell, we will means in the primitive cell, the planes have only all the points present at the corners. And in case of unit cell, the primitive unit cell is the one where all the points or all the lattice points, they are only present at the ends, or you can say the borders. So in the later slides, when we will be talking about all the four types of centering of the lattice points, then I'll tell you that there are different centerings, so how they vary. So primitive means only at the ends the points are present and not at the centers. And then comes 120-degree rhombus. So if we talk about this rhombus, so in this case, the A is equals to B, the distance between both the A and B are equal, and this theta is precisely equals to 120 degree. So now means by convention, it appears to be a hexagonal lattice, but somebody can also perceive it as a um, rhombus if we consider this particular plane, in which this is known as primitive cell because in this case there is no point in the center, whereas in this case we were having one of the lattice point in the center. So in this way, there are different types of crystals which are possible, and it also depends that how you perceive them. So in this case, this rhombus can also be perceived as a hexagonal lattice. Maybe someone can construct a hexagonal lattice and now they can accordingly calculate that what are the different symmetries, what are the operations that can be performed, different angles. So it depends on them. So this is how 120-degree rhombus operates. And then ultimately we'll talk about the last one, which is the parallelogram, in which case A and B won't be equal to each other, and whereas this theta angle, this won't be equal to 90 degree or 120 degree. So other than that, it can have any angle, which is general angle. In some cases, it is also known as rhomboidal lattice, or some papers also call it as oblique or monoclinic. So whenever you hear a word rhomboidal, so sorry, rhomboids, so that means a parallelograms with no 90-degree angles and two different side lengths. So in that case, these parallelograms are precisely called as rhomboids. So this shows you the parallelogram. Now we'll move to 3D lattices, which are our major interest. So in case of 3D lattices, we defined using the 3 basis vectors and 6 lattice parameters. So in this case, now the things, crystals, they will be in the 3 dimensions. So there will be 3 distance, because in the previous 2D lattice, we only have 2 distances, A and B. Now we will have 3 distance. So we will have A, B, and C. And now there will be 3 angles, alpha, beta, gamma. So previously there was only one angle between A and B. Now we have C also. So we will have 3 different angles. And there are some special cases which simplify the parameters, such as cubic. So in previous case, as we have square in which A was equal to B and alpha was 90 degree. So in this case also we have a special case which is of a cube. So cube is something like square in 3 dimension. So it has A equals to B equals to C and all its angles are equals to 90 degree. And then crystals consist of unit cells which are arranged in 3D space. So crystals come under 3D lattice. So there was a scientist named Auguste Brevis who has identified 14 different Brevis lattices, and these 14 Brevis lattices, then there are 7 crystal systems and 4 lattice centering types. So if we combine 7 crystal systems and then studying 4 different lattice centering types, then these 7 crystal systems will generate 14 different Brevis lattices. What it means that they will generate 14 different types of three-dimensional arrangement of lattice points simply. And the Brevis lattice, it is defined by the discrete translational operations and appears identical from any point within the lattice. So now we'll quickly talk about the 7 crystal systems which are present. So they are triclinic, monoclinic, hexagonal, trigonal, orthorhombic, tetragonal, and isometric. And based on each of these types of lattices, they will have, means because in this we will be talking about the A and B and C length. So in all of them, there will be some kind of variation either in terms of length of ABC or in the angles alpha, beta, and gamma. So with which they generate these different types of crystal systems. And now there are 4 lattice centering types, which means that at what position the lattice points are present. So in case of primitive unit cell, we have all the points which are only present at the corners. So if we talk about the primitive cell, it contains 8 into 1 by 8 equals to 1 lattice point. Now, why is it 1 lattice point? So, for each unit cell, we are calculating the lattice point. So, what is the contribution of the lattice point in each unit cell? So, in this case, if we have these, actually, we have these 8 lattice points, but all these 8 lattice points, each of these lattice point will be in the contribution with different other unit cell which will be surrounding it. So in that case, each of the unit cell will have 1 by 8 contribution. So that's why we have 8 lattice points. Each has 1 by 8 contribution. That's why we are, for a single unit cell, we only get the 1 lattice point. Similarly, for the case of body centered, we have one of the lattice point in the center of the body. And then we have 8 points at the, means you can say at the corners. So those 8 points for this particular unit cell, their contribution will be 8 into 1 by 8, which will be 1, plus this single point which is present inside the body. So it will completely contribute towards this single unit cell only, because it is not contributing to any other unit cell. That's why we are completely adding it. There is contribution of 100% by this one particular lattice point. So that's why there are 2 lattice points in case of body centered centering type. Then if we talk about face centered, then in this case, there are 6 faces which are present in a 3-dimensional cubic structure if we talk about. So in that way, contribution of the corner lattice points will be 8 into 1 by 8 plus these, because we know that each face, so for example, let's assume this particular face. So another unit cell which will be present adjacent to this. So in this case, this particular lattice point is shared among 2 different phases of 2 different unit cells. That's why its contribution will be half. So in this way, 6 lattice points present in the center of the, present in the face, they will contribute half. That's why it will sum up to 4 lattice points. Similarly, for side centered cell, so in some cases you will also see that instead of calling it as side centered, some people also call it as base centered, in which we only have the 2 sides, or you can say 2 bases, bottom and top. So in this case, the both of them also contribute half, because one unit cell can be present on the top. So this lattice point can contribute to the topper unit, the unit cell present at the top versus unit cell present at the bottom. Similarly, for the uh, lattice point present at the bottom. So similarly, it will also contribute half to this single unit cell. So that's why there will be two lattice point contribution by the side-centered unit cell. So this is how different 14 Brevis lattices are formed, depending on the arrangement of these lattice points and what is the distance among two planes, or you can say distance among the 2 points on three of the directions. Yeah, so this is also in the detail if you want to read. So you can go through the slide. And then this shows that what are the different 14 Brevis lattices, because cubic can have different types of phase centering, sorry, different types of lattice point centering. So for example, it also exists in the primitive centering as well as in the, so I stands for body centered. So it can exist as body centered cubic system as well as face centered cubic system. And in this, we do not have the side centered cubic system. Whereas for tetragonal also, we have two types of centering. So primitive will be present for all of them, whereas the body centered, face centered, or side centered, that we need to look. So in some cases where you do not find the face centered or side centered, so it simply means that they do not generate some variation in the 3D arrangement. So we will only consider the lattice which are unique from one another. So if some lattice centering also generates the similar pattern, then we will not no longer consider it. So that's why these are the different unique combination of 3D lattices which are generated. So these are the 14 Brevis lattices, and this is the table which gives the detail of each of them. That, for example, in the cubic, so simple in all these cases means the primitive one. So simple cubic has A, B equals to C, and alpha, beta, gamma, they all will be equals to 90 degrees. But if we talk about the face centered, so everything will remain same. But what is the addition? That we have the additional lattice points present on each of the face. So on all the six faces, there will be points. And if we consider the structure for all of them, then also it will remain same. There will be no change in the difference in the length of these 3 points or angles. So it will still remain constant. Whereas in some other cases, you might see that body centered shows some other variation. And then if you consider the face centered one, then they might change in the ABC or angles. Okay, so this is for the detail. If you want to study, then you can go through them. Okay, so these are all the 4 Brevis lattices. Okay, so now we'll study that what is the symmetry and what are the symmetry operations and why we need to study them. So when we call symmetry, symmetry comes under the geometric operations only. Geometry means what is the arrangement. But when we call it as symmetric, so symmetric means that if we perform its spatial transformation, it will still leave the object unchanged. It won't change it. So spatial transformation or symmetric operations are rotation, reflection, inversion, or translation. So that is not changing any geometry of that molecule. It is symmetric. It will come back to that particular position by making some of the angular motions or other things. So we'll see. Symmetry plays a critical role in determining the molecular structures and properties. So its applications include optical activity, infrared and ultraviolet spectroscopy, and crystal structure determination. So in this case, the main application is the crystal structure determination. Okay, so now these are the kind of symmetric axes of symmetry and what are the operations which are getting performed. So in this case, I've only considered rotation. So for example, if I have a structure like a hexagon, so in this case, there is 60-degree rotation. Six-fold axis means if I rotate it by 60 degrees 6 times, then it will come back to its original form. So this is known as six-fold axis, or you can say the 60-degree rotation. Then four-fold axis means if I rotate something by four times by 90 degree. So how we calculate the angle? So simply in each of the axis, we will divide 360 by 4. So it will give me angle of rotation, which will be 90 degree. So which means in four-fold axis, I need to rotate something by 90 degrees 4 times to bring that into the original form from where I started. Similarly, for three-fold axis, the angle of rotation is 120 degrees. And for two-fold rotation, the angle will always be 180 degree. And now it depends what is the crystal you are considering. And for example, if you are considering some crystal which does not have three-fold axis, means even after rotating it 3 times, you do not observe the similar uh, structure from where you started. So it depends that what is the crystal which you are observing and what is the symmetrical, means what are the symmetrical operations that can be performed, whether it is two-fold axis, it has three-fold axis, or two-fold or six-fold axis. So that's why we need to study them. Okay, so now as we have discussed that the unit cell is the smallest unit and it combines together to form the crystal lattice. In crystallography, we are looking at these crystal structures at molecular levels, and symmetry in crystals is important to define the crystal systems and unit cells so that we do not have to repeat certain operations. If we know that these two units are exactly identical, then instead of rotating it at redundant angles, we know that which angles can generate diversity. So we will only rotate our crystal at that particular angle only. So now crystals are classified based on symmetry and not just unit geometry. Okay, so depending on that, what are the different operations which can be performed on them, they can be, means those crystals can be classified on the basis of that. So symmetric operations, they include mirror, which is reflection, center of symmetry, which means inversion, rotation, and glide. So one by one, we'll go through these operations also. And then comes point groups. So point groups are the groups which categorize the crystals based on symmetrical elements. So in the later slide, we will also see that how many point groups are also possible. So from 14 Brevis lattices, we will see that how many point groups can be possibly produced depending on the symmetric operations. So after performing each symmetric operations, the arrangement of the lattice can change. So in that way, it will generate multiple point groups. So in this particular figure, I have shown the symmetry elements. So means depending on the which symmetric operation is performed, there are different elements from where you can perform the analysis. So first is the plane of symmetry. If you are considering this plane and then checking what is happening after symmetric operation, so this is known as plane of symmetry. Then comes axis of symmetry, which is passing from the center. So this tells you the angle of rotation, at what angle the crystal is being rotated. And then comes center of symmetry. So these are the points from where the other lattice points, they will be equidistant to one another. So from this point, all other points will be equidistant to this particular point. If we calculate the distance between this point and the topmost point to the center of symmetry, then these all will be symmetric. Their length will be equal. And if I cut it from any of the direction from the center of symmetry, so it will divide my crystal into equal halves. So this is what the center of symmetry means. And then symmetric operations are translation. So in this case, this is just to show you that this is a 7 kind of a thing. So after translation, it has moved by some distance and it has reached this. So, but it is identical. The difference is only in the position. If we talk about reflection, so if we see something in the mirror, so this is the mirror image of 7. And by rotation, we mean that we have moved it by some angle. So this is showing the example of rotation. And in case of inversion, it is inverted. It is flipped, you can say. So this is the example of inversion. Yeah, so now we'll quickly see the symmetry operations. So this is the reflection. So I'll also give you the idea of the coordinates, that how they will change. So reflection means in which the set of points, they will be mirror across the plane. So in this case, they will appear as the mirror image of each other. So when we say mirror image, which means that as soon as we try to superimpose them, they will not superimpose on each other. So like our left and right hands, similarly, these are mirror images. So in reflection, we have the mirror axis. So from mirror axis, each point which is present on the original object versus the reflected object, their distance from the mirror axis will be identical. So this point from mirror axis and this point from mirror axis will be identical. That is A. Similarly, these two points will also be completely of identical length from the mirror axis. So in this case, we are calculating the distance from the axis. Now, what is happening to my x, y, and z points? So x, y, z are the three axes. So in this case, if the mirror axis is perpendicular to x direction, then there will be only change in the x-axis. So now my x-axis have become negative, whereas my y and z-axis, they are still the same. So reflection is only changing one of my axis, and that axis is the one which is perpendicular to the mirror axis. So for example, you assume that this is my x-axis, um, this is my topmost, means towards the top, or the vertical axis is my y-axis, and the one which is projecting outside, that will be z-axis. So if I compare both, means three of these axes, then there is only difference in the x-axis. So previously it was towards the positive, now it has changed to negative side, and rest other axes, they are still the same. There is no such difference. Okay, so in this case, you only have to remember about the mirror plane, and the distance from each point will be equal. They will be non-superimposable. This is about the reflection, and only one of the point or one of the coordinate will get changed, which will be perpendicular to the mirror plane. Rest other points will remain constant. These are the points which you need to keep in mind when you're talking about the reflection type of symmetric operation. Now, second symmetric operation is the rotation, where you are rotating the crystal by some angle. So we call the particular point from where we are rotating as the axis of rotation. So this is the axis of rotation from where I am calculating the angle. So in case of one-fold rotation, so one-fold means if I rotate something by 360 degree. So whenever I rotate something by 360 degree, it will come back to its original form. So there is no difference between both the original and the rotated form. So they are exactly identical. Whereas if we rotate it by 180 degrees, so this comes under twofold rotation. That means I again have to rotate it by another 180 degree to bring it back to the original form. Similarly, in case of threefold also, I need to rotate it three times to bring back it to the original form. Similarly, if it is fourfold, then 90-degree rotations need to be performed four times. In case of sixfold, 60-degree rotations need to be performed six times. And then in case of, you can say, the line land where we have infinite points, so this has the infinite rotational. So in this case, nothing will, you need to perform, if there are infinite points, then you need to perform infinite rotations. So this is just to give you an idea that if there are infinite points, then what will happen. Okay, so these are the different symmetric operations, and depending on what are the shape of the crystal, it depends that what could be the number of fold rotations which will be applied on them. Means which type of symmetrical operations will work on them. So then comes the third one, which is the inversion. So inversion means it is a symmetric operation which pulls every point through an inversion center. So in this case also we'll have an inversion center. So in rotation we have the rotational axis. So in case of inversion we have the inversion center, and now these two images, they are inverted. That means the points which are present, they are in opposite direction to one another. So in case of inversion, you can see that the previous coordinates were x, y, and z. In case of inversion, all these points have changed. So they are minus x, minus y, and minus z. So if there is inversion, then keep this thing in mind that all the coordinate points will change. If they were previously positive, now they will become negative because everything is changing. But the same, but how it is symmetric? So if there is some inversion center, so from there, the distance of the identical points, these both are just the inverted, means inverted operation has been performed on this particular crystal. So in this case, these are inverted forms. So if we calculate the identical point in both these forms, so this one and this point here, so from the inversion point, their distance will be equal. The distance means their length will be identical from the inversion point for all these points which are present in the crystal. So this is all about the inversion operation. And then comes translation. So translation means if something moved by some distance. So this is a symmetric operation that moves a set of points through space. So as you can see that this figure, this particular crystal has moved by some distance and it has reached here. So in this case, point groups do not involve translation. Any point x, y, z becomes x plus a, y, z. Now why x plus a? Because translation is happening in the x direction. So it depends that in which direction translation is happening. So whatever direction the translation is happening, you need to add something, means at what distance that translation has happened. So similarly in this case, if translation has happened towards the direction, then we will add some distance a by which this movement has happened. So similarly, this is how the coordinates positions will change for that particular crystal for which we are studying the symmetric operation. So this is translation. And now the last one is glide. So glide is a symmetric operation which is not a single symmetric operation. It combines two different symmetric operations. So one is translation and another is reflection. So in some textbooks, glide is also referred to as transflection, which is a combination of translation and reflection. So what is happening in this case? Firstly, translation is happening. So crystal is moving by some distance and then it is getting reflected. So there is a generation of a mirror image. So now this is the original crystal, and after the glide operation, this is the means after the symmetric operation, the crystal which is obtained. So this is known as glide. Okay, so this is the glide plane, and then from glide plane, if we calculate the distance of each point, so that distance from the plane will be equal for each and every point among both these crystals. Yeah, so in this case, you only need to remember the glide plane and then about the combination of translation and reflection. So this is about the glide. And now this is to show you a little bit about mathematical representation. So for example, if we consider 180-degree rotation around the z-axis, so as we have seen in case of rotation, that whenever a rotation is happening, so it depends towards which direction the rotation is happening and which axis is kept constant. So in this case, as you can see, this is the z-axis and the crystal is moving. So this is just a representation in which the wheel is moving across the z-axis. So z-axis is fixed. Now it is moving, and x-axis and y-axis are changing. So in this case, z coordinates remain unchanged while we are only flipping the x and y-axis. So now x and y-axis have become -1, whereas z-axis is fixed to 1. This is just a representation that how we can represent it in the case of metrics. And then types of rotations in crystals. So this we already have discussed, one fold, two fold, three fold. So it depends that after how many fold rotations by what particular angle, we will get back to our original crystal, from which we have started. So this is it about the types of rotations. Yeah, so now we'll talk about the space groups and crystal systems. So now, till now we have seen that there are 14 Brevis lattices and there are 7 crystal systems which were cubic, tetragonal, orthorhombic, hexagonal, trigonal, monoclinic and triclinic, and this is the representation. So these come under 3D lattices, three-dimensional shape where they will have three distances A, B, C and three angles alpha, beta, gamma, and depending on the variation among these 6 values, there are 7 different crystal systems which are possible. And then 14 Brevis lattices, which also we have seen that. Then comes centering. So on the basis of centering on these 7 crystal systems, we generate 14 Brevis lattices. And after performing the symmetric operations, we can generate 230 space groups, so which describes complete symmetry of crystals. So in this way, if we know means about all these space groups, then we can decide that what is our crystal shape, what is the symmetry of our crystal, and then we can judge that at what angles we can perform the rotations, which will generate the unique structure of the unique three-dimensional arrangement of the crystal, so that we can maximize our results which are generated. So this is the reason why we need to study the symmetry in crystals and what are the crystal systems which are available. Yeah, so this is just to sum up all the things which we have seen so far about the crystals. So crystal systems, we have Brevis lattices, we have seen point groups and space groups. So there are total 7 crystal systems. And if we perform translation, centering, all these things based on the phase centering or translations, then there is a possibility we can generate 14 Brevis lattices. And if we perform the symmetric operations on these 7 crystal systems, such as rotation, reflection, inversion, glide, then we will get 32 point groups for the 7 crystal systems. Now, if we combine all of them, including glide, screw, so screw is also one of the symmetric operation only. So we can generate 230 different space groups. So these 230 space groups are unique from one another. So this is all about the different 3D lattices which are possible by performing all these operations. And this slide is about the significance of symmetry in crystals. So they define point groups, which helps to classify crystals. Then X-ray diffraction patterns, because it will also affect the X-ray diffraction pattern. What is the spacing among two lattice points can also determine that at what angle the light can get X-rays can get diffracted. So in that way, this understanding the crystal and means understanding all these things are quite helpful, and it also determines the physical and chemical properties of the materials. So in order to conclude it, symmetry is fundamental in crystallography, which helps in understanding the molecular arrangement and properties of molecules. Combination of symmetry operations define the possible crystal systems in the or crystal structures in the nature. Okay, so now we will go through the X-ray crystallography data collection. So this was all about the crystal thing. And previously we have started with the source of X-rays and X-ray detectors and everything we have covered. So now the step is collecting that data. So after the diffraction pattern is generated, how to process it, how to collect it, and how to store it. So now we'll study about that. So is it clear till now? Okay, thank you. So now we will start with the um, what is the X-ray diffraction pattern and how it interacts with matter. So diffraction pattern, it results from the interaction of X-rays with a single crystal sample. So there is a single crystal which is mounted over the goniometer, and now X-rays are passing through it. So now these X-rays, after passing, they generate some diffraction pattern which is collected using the CCD detectors with the goniometer angle set to 0. And the pattern consists of distinct spots corresponding to scattered X-rays. So those spots are obtained at those sites where the diffracted X-rays are getting scattered to. So in that way, they are telling you the positions of lattice points in the crystal or position of atoms inside the crystal. So interaction of X-rays with the matter. Now, how the X-rays are actually working. So there could be coherent scattering, which is the Bragg scattering, and which is important for the diffraction data, because if the scattering is coherent, then they will sum up and the intensity of the wave will increase. Incoherent scattering, also known as Compton scattering, so it produces noise because these X-ray waves or these waves which are generated, they are not consistent. They are kind of a little scattered and they are not consistent. So they are also known as incoherent. And then absorption and fluorescence. So it contributes to energy loss because if there is absorption of X-rays or if there is fluorescence generation, so there will be some loss in energy. Whereas photoelectron emission is the secondary effect, which is generally not utilized in diffraction. Yeah, so this is to show you what constructive and destructive interference means. So even in case of coherent this coherent scattering, there is a possibility that you might observe constructive interference or destructive interference. So if two waves, they have the means means if the you can say the amplitude, if amplitude and the distance, this wavelength, if it is at the constant position, then they will sum up if they are in different phases. So this is just a phase, that if it is in different phases, then there will be destructive interference if they are in the same phase, or you can also call it as a in phase. So if two waves are in phase, then they will sum up and it will be a constructive interference, and which will form the diffraction spots. Whereas if they are out of phase or they are not in the same phase, for example, in this case, it is negative, whereas at the same position, it is positive. So they are in different phases. So they will cancel out the effect of each other, and this will generate a destructive interference, which will prevent the signal formation. So for our case, we want that the waves which are generated, they should be coherent and they should be of in phase, and there should be constructive interference. And then another thing is the scattering angle. So there should, if there is a higher scattering angle, it results in decreased coherent scattering. So we want that the scattering angle should also be less. It should not be high. So this is to show you that what will happen if the coherent angle is large. So if it is 2 theta, then what is happening is that it results, so coherent scattering, it results um, from the combined scattering of all its electrons with phase differences due to their spatial positions, leading to reduced scattering intensity at higher angles. So if means when we say scattering, so we want that each electron, it is the sum up of the angles at which the X-rays are diffracted by each single electron. So if this angle will be quite large, then we won't get many such radiations which are getting diffracted back. So in that way, we want that the angle of this diffraction or scattering angle, it should be low, so that multiple such scatterings could be observed. So that's why we want lower scattering angles to get coherent scattering. And this is to show you the difference among diffraction and scattering. So when we say scattering, it is the random interaction of X-rays with the single atoms, and they can get randomly scattered in different directions. But if we talk about diffraction, then it is the ordered scattering from crystal planes. So there is some order, there is some direction associated with it, producing strong signals in specific direction. So this is not randomly passed on different directions, but it is precisely passing in the specific direction and which is coherent, which is ordered. The distance between each such wave which is passing, it will be at this identical length, or you can say it will be of ordered nature. According to Bragg's law, the diffraction condition is n lambda, which is the wavelength, it will be equals to 2d sine theta. So depending on the distance among two unit planes, which is d, so in two planes, the distance among lattice point is d, and then sine theta. So this tells you the wavelength, which we have seen previously. So if this formula applies, that means you will be able to get better diffraction pattern. And then comes the data collection process. So first of all, we start with crystal preparation and then initial alignment. So which ensures that alignment here means that how you are aligning the crystal on the goniometer. So if that alignment is proper such that the X-rays can pass through the center of the crystal, then it is better. But in case you're not aligning that, means using the cryo loop, you pick up the crystal and align it on the goniometer tip. So if that alignment is not proper, if there is some rotational or there is some translational errors among different layers of that particular crystal, then it will generate some errors. So we need to make sure that alignment is proper. So in this case, we are ensuring that there is accurate positioning of the crystal. Then comes detector distance and rotation. So it is set for the optimal diffraction capture. And then oscillation images. So after each oscillation, because we are oscillating across different angles to get different types of crystal lattices, so collected at small angular increments. So means we change angle by smaller increments to check that whether there is some change in the 3D lattice structure which we are observing, or lattice points. And then there comes automated indexing. So after doing all of this, we means does the automated indexing. So we use certain software which analyze those images or the diffraction patterns which are obtained to determine the crystal parameters. So because after crystal parameters have been determined, so we will be able to know that what is the shape of that particular crystal and what angles we need to further move, means what are the other oscillations we can perform. So this is about the data collection process, and further data analysis that will be covered in your next week. So this is just a representation that how from crystallization we move till the atomic model. So till now we are only at the data collection level. Then comes data analysis, and then after data analysis we also have to refine it so that proper atomic models could be generated. So because in X-ray crystallography we also face the phase problem. So we also need to deal with it, how to means deal with the phase problem, how to avoid it, because we are till now we are only getting the intensity information of the X-rays which are getting diffracted, but we do not know at what angles they are actually getting diffracted. What is the phase information? So in the later sessions or in the week 5, you will see that how we will deal with the phase problem, and then ultimately how the data is uploaded to PDB or other data sets after modeling and refinement. So if we talk about indexing and data processing, then they determines the unit cell dimensions. So for each crystal, unit cells are identified, what is their dimension, what are the lattice types. So lattice types means what is the 3D arrangement. So cubic, monoclinic, so that is the lattice type, and its orientation, that at what angle it is oriented. And common softwares which are generally used for indexing, they are HKL and MOSFIL. And refinement steps. So after indexing, we need to perform certain refinements after obtaining that diffraction image. So collection of initial diffraction images, integration of reflection intensities. So till now we have obtained the reflection intensities, and then scaling, merging and averaging of diffraction data, and computational structural refinement using the chemical constraints. So these are different steps under refinement. And then we'll talk about the reciprocal lattices or the Miller lattices and the Miller lattices, sorry. So reciprocal space representation. So now there is a real space, which means the real crystal. And then comes reciprocal space or the reciprocal lattice, which is the reciprocal of that real space. So we need to perform it because during crystallography, there is the inverse relationship between the diffraction pattern which we obtain. So that diffraction pattern is not the exact same as the real space, it is the reciprocal. It is like a reciprocal. So that's why we need to perform this. We need to represent it in the form of reciprocal space, for which we use the Miller indices. So Miller indices HKL, they define the crystallographic planes in the unit cell. So because now we are reconstructing what is in the crystal, because we are visualizing its structure using X-ray crystallography. So now we are making that unit cells, we are visualizing it. So Miller indices are generally used to define those crystallographic planes, and it used to assign the diffraction spots and measure their intensities. So this is the crystal structure which has a crystal lattice in the real space and the unit cell content. So this shows the unit cell and its means different distances and the different planes. And then if we talk about the diffraction pattern, so it will also have a structure factor, and it is a reciprocal lattice, which is means we need to calculate the reciprocal of all these uh, points or all the distance which are in the unit cell. So now we'll see how the Miller indices are calculated and how we can perform the reciprocal calculations. So when we say Miller indices, this is a notation system which is used in crystallography to describe the planes in the crystal lattice. So these planes, they are defined after getting the diffraction pattern. So at that time we use Miller indices, and these are the set of 3 points. So previously we were having the A, B and C distances, but in this case, Miller indices, we have H, K and L, which denotes the orientation of a plane with respect to the crystal axis. So if these are crystal axis, then what is the orientation of plane? So in this case, if we consider it as first position, second position and third position, so the position which is connecting to this plane, which will get the value of 1 and others will get the value of 0. So in this case, means whichever position is getting connected or which is having some attachment to this plane, it will get the value of 1. So in this scenario, as you can see in this case, the A1 is getting attached, is hitting at the point of this plane, so it is getting 1, whereas others are 0 0. In this case, the second position is sharing the lattice point with this unit plane, so it is having 1 value, whereas first and third position is 0 0. Similarly for other cases as well. So the key characteristics of this Miller indices are they are represented as HKL, where HK and L are integers. So they are integers. They could be both negative, positive, or whatever value is possible. And they are defined as the reciprocals of the intercepts, but most probably they will always be positive only. So defined as the reciprocals of the intercepts that a plane makes with the crystallographic axis. So in the later slide, we will see that how it is calculated. So that's why it is defined as the reciprocal of intercepts. So we'll calculate the intercept and then perform their reciprocal, and some other calculations will be done to find the value of HK and L. So it is used to describe the lattice planes that pass through the lattice points. So these are the lattice planes which are passing through these lattice points. So this is the lattice point. And then planes with the similar indices, they are parallel and equidistant in crystal structure. Yeah, so now I'll show you that how we calculate it. So procedure to determine Miller indices are, firstly we need to find the intercepts. So intercepts of the plane along x, y and z axis in terms of lattice constants a, b and c. So the plane which is defined, we'll see that where are the intercepts. So along x, y, z axis where my plane is cutting. So that will be my intercepts, and then take the reciprocal of these intercepts, multiply them by the least common denominator to obtain the integer value, and write the final indices in parentheses HKL. So in this case, y integer value, because this can go negative in cases of x, y and z axis, and there is some rotation, then the values present in x, y, z axis, they can also be negative. So in that way, if you will be performing these calculations, then in some cases you might see that HKL value can also be negative. So this is one of the examples. So in this case, we have x, y, and z planes. So along x, y, z planes, x, y, and z axis, we will see that this is the plane. So it is cutting here, here, and here. So we will calculate the intercept. So these are the intercepts where these points are cutting at the x, y, and z axis, or you can say where they are connected to x, y, and z axis. So in this case, towards x-axis, the intercept is at point 2. So if we consider it as 1 and this as 2, so this is 2. Along y-axis, it is it means cutting at point 3, and at z-axis it is cutting at point 2. So first step was to find the intercept. So now the intercepts are 2, 3, 2. Second step is to take the reciprocal. So now reciprocal will be 1 by 2, 1 by 3, and 1 by 2. Now we need to find the least common denominator. So if we find the denominator, then it is 6. So we need to multiply each of these reciprocals with this least common denominator. So 1 by 2 by 6 will be equals to 3. 1 by 3 into 6 will be equals to 2. And similarly, 1 by 2 into 6 is equals to 3. So now this is how we will calculate our Miller indices, which are H, K, and L. And these Miller indices, they play a crucial role in determining the crystallographic properties, including the cleavage planes and X-ray diffraction patterns and material behaviors. So this is about the Miller indices. And finally, in the data analysis, so diffraction data is processed to determine the electron density map. Resulting atomic model is stored in the protein data bank, which is the public database. So whenever the structure is resolved, then it has to be uploaded so that others can also access it. And then we can perform later on interpretations, such as on the basis of its functionality, its biochemical properties. Because if we know the structure of the protein, then we will be able to know that whether it is what is, if we have the idea that it is an enzyme, then with the structure we will be able to know where are the binding pockets present, how the different residues are arranged, which are at the surface, which are present at the core. So these are certain analyses which we can later on do. So in this case, I have gone through a review in which they have shown that how the structural determination of a protein is very significant in contributing towards the disease and drug analysis. So we can understand the disease as well as we can also generate drugs for them. So in this case, if we know a disease, we can also identify the targets. For example, from a diseased person, we can collect the samples, and from those samples, we can also get the proteins. We can visualize them, that what are the proteins, uh, how they look, and then after identifying the target, we can determine its structure. And now because artificial intelligence has come up, machine learning has come up, so there is a possibility that we can also predict the structure, such as AlphaFold. So by using that, we can also predict the structure for this target which has been identified. So predicted structure and experimental structure can be compared. Experimental validation can be performed for the predicted structure, and then we can do lead screening. So lead simply means the protein which is like responsible for certain disease, so that act as a lead, and then target lead structure. So among that particular lead is a bigger molecule, so in that we need to find the target where we can for which we design the drug. So not for the entire protein, we need to find certain region in that particular protein molecule where we can, I mean, for example, if we know the surface residues which are charged in nature or which are majorly involved in interactions, then for that particular site, we can design some small molecules which can bind to that site. So in that way, target region is identified and then drug candidates are designed. So then clinical trials and ultimately drug is designed. So this is just to give you an overview that how the structural identification can help you to design the drugs and identify the proteins which are involved. Along with this, physicochemical properties can also be identified of that protein, and other analysis can also be done. Yeah, so this is the review. If anyone is interested, then you can go through this. And this was published in 2023. So it tells you about the current role and evolution of X-ray crystallography in terms of drug discovery and development. So you can see that how it has played a significant role in X-ray crystallography, although we have seen that cryo-EM is also available, NMR is available, and other techniques are also available, but still X-ray crystallography is still the golden standard. It is still the better among all these techniques. But the limitation is just that we are not able to generate many crystals for certain proteins. It is time consuming process, and as I always say, that it is the art. So sometimes you might get the protein, sometimes you might not get. So in that cases, it is a time consuming. Otherwise, this method is highly confident. So for structural analysis, mostly people prefer X-ray crystallography. And then in this paper, in this particular review only, they have, when they were performing multiple comparisons among different studies, so they have shown that if we compare the structures among X-rays and cryo-EM, then many structures have been solved by X-ray crystallography than the cryo-EM. And then if we talk about the resolution, that how well we can distinguish among 2 points in the 3D space, then the X-ray crystallography has a better resolution, whereas cryo-EM has a little lesser resolution. So whenever we compare resolution, so we only look at the value which is less. So in this case, X-ray has 2 angstrom of resolution, whereas the cryo-EM has 3.4. So resolution value which is lower, that is best resolution. So in this case, X-rays have higher resolution. So in this way, they have mentioned that why X-rays are better, X-ray crystallography is better. So now we will practice few MCQs. So is it clear till now? Okay, so maybe you can go through these things again and again. Then you will be much, means understand more and more these terms. Otherwise, with the first go, it is quite difficult if you do not have any background in this field. So don't worry, means you will get through it whenever, means in the next session also, when new topics will come, somewhere you will, these basics will also come again and again. So means till the end of this course you will have a better understanding, so don't worry. Now we will practice these MCQs. So if you have any idea, then you can answer in the chat box. So the first question is, wavelength suitable for X-ray diffraction application ranges from, so option A is 0.5 to 2.5 nanometers, option B 0.5 to 2.5 angstrom, option C 0.5 to 2.5 micrometer, and option D is 5 to 25 nanometers. So do you remember? Okay, option B. Okay, now let's see. So what is the right option? Yeah, you're right. So the right answer is 0.5 to 2.5 angstrom, because in the initial slides only we have seen that for X-ray diffraction it is generally between 0.5 to 2.5. The second question is, in small molecule crystallography, which of the following source is used? Copper, palladium, silver, and molybdenum. So this also we have discussed in the initial few slides. Yeah. Very nice. So the right answer is option D, which is molybdenum, and it has K alpha of 0.709. So, sorry, it has the lambda absorption value for this radiation is 0.709. So that's why it is less than the copper. So it is generally used for the smaller molecules. Now, M to K shell transition of an electron generates which of the following emission? K alpha, L alpha, K beta, or K mu? Okay. Yeah, so you're right. It is option C, which is K beta. So this also we have seen previously, that whenever the movement will happen from L to K, electron will come from L shell to K shell, then it will be called as K alpha. Whereas if it comes directly from M to K, then it is K beta. Okay, so next question is, which of the following is not a symmetry operation? Option A, inversion, option B, translation, option C, rotation, and option D, diffraction. So which among them is not a symmetry operation? Yeah, so this is also right. So the right answer is diffraction, because rest others are the symmetry operations, inversion, translation, rotation, as well as reflection and glide are also among them. Okay, so now let's move to our fifth question. So which of the following represents the twofold symmetry, twofold rotation symmetry? Solid oval, solid square, solid pentagon, and solid hexagon. So now you need to tell that Which of them has twofold rotation symmetry? Twofold symmetry means if we rotate it by 180 degrees two times, then it will come back to its original form. Okay, so now let's see. So the right answer is solid oval. Okay, so it's not solid square, it's solid oval. So because in square we have four different sides where we can have other points. So by 180 degree we won't be able to complete it. We need it is having the fourfold rotation, because after the fourfold rotations we will be able to get it back to the its original form. So the solid oval is the right answer for this twofold rotation symmetry, and this is how, by rotating it by 180 degree, it will come back to its original form. Okay, then comes the next question. So the symmetry operation glide plane consists of option A inversion and rotation, option B translation inversion, option C reflection and translation, and option D is rotation and reflection. So this is about the glide. Option B is translation and inversion. Okay, now let's see. So the right answer is reflection and translation. So in this case, firstly it translates, or you can call it either way, means it depends. So in this case, firstly it translates and then that translated thing gets reflected. This is reflection, this is not the inversion. Or means other option could also be translation plus reflection. So they both are same. These two symmetric operations, they combine together to form another symmetric operation, which is glide. And remember it in the form of the person who is in footsteps on the beach. So they are like the mirror image of each other. They are not inverted, they are mirror image, and they are different from one another's position by some distance. So that is also one of the best example of glide. So reflection plus translation. Now the next question is, a unit cell has a shape of 120-degree rhombic prism. The unit cell will have which of the following crystal system? Option A, cubic, option B, hexagonal, option C, monoclinic, and option D, triclinic. So this question is, I guess, quite difficult. Okay, now so let's see. Yeah, so the right answer is option B, which is hexagonal. So as we have seen in the previous slides also, so if we talk about the 120-degree rhombic prism, then other perception of watching it could be a centered hexagonal in which there is a point in the center. So it obtains its unit cell is can also be hexagonal. So this is that's why it is hexagonal, and it can't be cubic because there is no possibility of cubic. Then monoclinic and triclinic is also not possible. So hexagonal is the one which is possible in the case of 120-degree rhombic prism. Okay, so next question is, which of the following filter is used to reduce K beta X-rays from copper anode? So this was covered in your means this week session topics. So I haven't included this slide in my presentation, but I hope you might have some idea about it. So option A is nickel, option B is cobalt, option C is iron, and option D is lead. Okay, so according to you, lead is the right answer. Okay, so the right answer is nickel. So in case of copper anode, we use the nickel as a filter. So when we were talking about monochromators, so in that case, we were also studying that monochromator role is to allow the specific means wavelength of light to pass through it. So with that specific wavelength, we were focusing on the means K alpha radiations which are passing, because K alpha are high intensity radiation. So we want to avoid the noise. We want that K beta does not pass and only K alpha radiations pass through. So for that, in case of means other than monochromator, we go for the interference filters, in which nickel is the best, which can filter out the K beta radiations and which only allows the K alpha radiations to pass. So it will increase the intensity of K alpha and the K beta intensity will be diminished. So that's why this is the best filter for copper anode. And for other types of anodes, the choice of filter will vary. Now the next question is, which of the following metal is used in liquid state in X-ray sources? Copper, mercury, sodium, and gallium. Yeah, so this thing also I have not discussed today, but it has been discussed in your week topics. So any guesses for this? Which of the following metal is used in the liquid state in X-ray sources? So I'll give you a little bit of a hint that because in this case we are generating X-rays. So till now we have seen that what is the limitation of different X-ray sources, that whenever that electron was coming from cathode and it is hitting the anode, then there is a generation of heat. So to avoid that, in case of rotating anode, we have seen that it is continuously rotating. So that's why the heat is spread across the entire area. So similarly, to avoid that thing, to avoid the heating up, because in the rotating anode also we have seen that there is wear and tear on the um, means the back of means the rotator which is rotating it. So to avoid that, what else can be done is that we can use the liquid metal. What it will do, that on that anode surface it will bring the new liquid metal on the top of that surface. So whatever surface is wear or tear, so the now the another metal which is coming in the liquid form, so it will replace that surface so that not much heat is dissipated at that point. Okay, so right. Yeah, so the right answer is option D, which is gallium. So now next question is, generally the protein crystals have a big lattice, so the spots obtained for such crystals are option A, very far from each other, option B, relatively far from each other than the small lattice crystals, option C, very close to each other, and option D, none of these. So now you have to tell if the lattice is big, then what will be the sports? What will be the distance among them? Okay, so let's see. So what is the right answer? So the right answer is very close to each other. So if you have a big lattice, then the spots which will be obtained, they will be very close to each other. So these were the questions which were discussed or which were present in your previous year assignment. So means keep this thing in mind that whenever you will be preparing for your exams, then do go through them once again because these might come in your final exam. So this was all for today's session. If you have any doubts so far, then let me know, or if you need any study material or anything else, then please let me know and then I'll end the session. Okay, so actually, I'm not currently going through the discussion forums, but I will try to look for your queries at that site. So, for example, if you have any doubt regarding these topics or any questions, then you can post it on the Q&A section of the NPTEL. So in that discussion forums, it will be visible to all, so we can see from there and we can answer it. And then, okay, so I'll provide you the study material on lattice, and along with this, in your shared drive, I have also added a document which is about the people who have contributed in X-ray diffraction. So in that particular, that is a document, that is a PDF in which many important concepts were there. So if you are interested, then you can also read it. And about the lattice, so I'll provide you the document, which will be in detail. So I think this shared drive is accessible on the so whenever you go to the NPTEL this particular course, then in the bottom you'll see problem solving session. So in problem solving session only, when you click on it, then in the window which will appear, so in that only you will be able to access the Excel sheet where both the TAs, so me and along with me, there is also another TA. So she is also posting the um, means the content which we are teaching you. So this is the recorded session. So we have the link for the YouTube recordings, as well as we also share the drive where we keep all these presentations. Yeah, and if you are not able to access to drive, then in my even my YouTube videos also in description I put the link to that particular slides. So you can go through that way also. Yeah, there will be all the recorded sessions. Okay. Yeah. Thank you so much for listening. And for any doubts, feel free to comment on Q&A. So in that, anyone who sees it. So there are some official TAs as well, other than we two. So they will also be able to answer it. And even the sir, means the instructor will also be seeing the discussion forum questions. So he will also answer if there will be such questions. Okay, so thank you. And now we'll see you in the next session. So I'm ending the call.